Yes, so it's not only my opinion, it would be the opinion of the broader scientific community and clinical community who is involved in this. The National Cancer Institute of the United States of America, where I come from, convened in 2013 and 14 a large group of scientists with international participation. They asked them a simple question, which is a very complex question: what are the priorities in small cell lung cancer? And there are 5 priorities that were designated, and I believe these priorities still hold today. The first priority is to create better tools for the research of small cell lung cancer. Small cell lung cancer is a very difficult disease. Not only it's aggressive and we lose many patients around the world. In the United States, it's about 30,000 patients a year that are lost to this disease. But it's very hard to deal with. That means that there is a paucity of specimens. We do have some specimens, but it's by far not enough. So just to collect these specimens, to get access to these specimens. But at the same time, we need new models. We have many cell lines already, mostly created by Dr. Mina and Azdar's group, currently at UT Southwestern. But there are animal models that are still in need. So that would be a task to follow in the future. There are some models already available. There are gen models, there are PDX models, but it's still not enough. So that's the first need. The second need would be a better understanding of the genomic and even proteomic and maybe metabolomic outfit of small cell lung cancer. Again, studies were already conducted. There are several hundred patients that are currently sequenced. However, that's not enough, specifically in a disease like small cell lung cancer, where it's known that it's caused mostly, in most of the cases, by environmental factors such as tobacco use, and the mutational rate is very high. And to have such a tumor genetically or genomically classified, you need many specimens. Some people say you need maybe 1,500 specimens to get a good grasp on what. So we are not there yet, and that's definitely a challenge, and that needs to be done. The third priority would be early detection. Now we know, and prevention. Now we know that smoking is the major culprit, but how to detect in these high-risk populations, like people who smoke 20, 30 pack years, how do you follow up, how do you detect this disease? It's very hard, it's not easy. And these studies are very tedious to do because you need to have very large cohorts that you can follow over the years. And then you have maybe several tens of thousands of patients where maybe 40, 50 of them would develop eventually small cell lung cancer. And then you can look at serum or blood-based markers. So that's a very difficult task, very demanding on the number of patients, number of people, subjects to screen. So that's the third. The fourth priority, and that's perhaps the most advanced currently, is focusing on therapy, new therapy developments. That means finding new targets, which is difficult, but there are already some clues. And perhaps most recently looking at the potential of immunotherapy. And the last priority would be also related to therapy, but trying to decipher why there is this resistance that develops in small cell lung cancer, and what are the mechanisms, how to overcome that. So those would be the 5 priorities that are currently still important for investigators and clinicians to deal with. Yes, I'm happy that you asked this question. We are very excited about this new initiative, which took several years to put together. We solicited applications, and the applications receipt dates are still open. So since last year, we already collected many applications, and then we selected the best of them. And we will have another solicitation this year in November, another 2 dates next year in March and November. And we really encourage international groups to join this. So for now we have 6 projects. In the abstract books, I was able only to reveal 4 of them because the other 2 were not funded yet, but currently they are funded, so I'm going to talk about those. 2 of these projects are related to the TOUGH program, the early detection. There are 2 groups. One of them is at in Virginia Commonwealth University, and another one is at the MD Anderson Cancer Center in Houston. The first group, led by Patrick Nanasingham and his colleagues, is devoted to really brand new cutting-edge technology where they focused on so-called exosomes. These are small particles detected in blood that are derived from cancer cells. And they are able to detect the content of these exosomes, mostly mRNA or microRNA, practically RNA. And what they are doing, they have special technologies where they create these platforms with nanoparticle based, and they are able to detect minuscule amounts of RNA in these exosomes. And based on that, they hope to be able to create a very inexpensive yet powerful tool how to contribute to the early detection of the disease. Dr. Hanashi, Sam Hanashi's group at MD Anderson, is also looking at blood-derived biomarkers, but they are more interested in proteins and in antibodies. So that focus is solely on that. And there is some good preliminary data that maybe there is some information that can be extracted from the blood to be able to early detect small cell lung cancer. The other projects are more related to therapy. So we have Dr. Poyer's and Charlie Rudin's group, who is in fact the head of the overall consortium from Memorial Sloan Kettering. They have developed already in the past, a recent past, a very interesting drug against DLL3, which is a surface molecule that can be readily targeted with an antibody. And in the past, they used an antibody linked with the toxin that was able to be very actually successful in early phase clinical trials. However, there was some toxicity related to those trials. So what they are trying to do now, they are creating a so-called theranostic preparation, which means there is this antibody against DLL3, but it's linked to isotope, so radiative isotope, which means they can actually not only treat the patients, but they can actually detect where this antibody is homing, and that's very important for the quality control, let's put it this way, and even diagnostics. So they are going that direction. Then there is another group, again at MD Anderson and also at Stanford. These 2 places teamed up to put together an application where they look obviously at immunotherapy, but a combination of immunotherapy with drugs that are directed to DNA repair. So the combination seems to be quite promising, and they are trying to improve the current results with immunotherapy in that regard. There is another group at UT Southwestern, Dr. Mena and his colleagues. They are now they are looking also at therapeutic targets. However, in their case, they're focusing on two defining molecules that basically define the neuroendocrine nature of small cell lung cancer. ASLC1 is one of them, and EuroD1 is the other one. Those two are observed to be expressed in small cell lung cancer in such a way that they're most, I think it looks like more likely that part of the small cell lung cancer specimens have one of them, the other part has the other one. And what they are trying to do is to not only study these molecules, their expression, but also look at the pathways they control and whether there are some downstream targets that could be then utilized for therapy. I believe I have already covered all of those that I wanted to cover. If not, I have to go back. So, as I would just relate to the first answer that I have given. So, first and foremost, there is really a paucity of specimens. It's very hard to get small cell lung cancer specimen in enough volumes, but also the number is important. So that's a challenge for most of these projects. There is also obvious challenges with new technologies like Dr. Nana Singham's project, where they are in fact developing the assay as they go. So there's a lot of improvement, and there are many technical problems with that. That's why we feel that a consortium that would bring these people together, perhaps scientists that look at different aspects of small cell lung cancer, would be able to work together, join resources, and that's what's happening now. You're trying to join these resources, make models available, create an exchange where everybody would bring in projects. I didn't mention that besides our funded projects, we embraced within the consortium projects that are currently funded through the National Cancer Institute but are independent or used to be independent from the consortium. That means that there are several investigators that look at resistance patterns and so on and so forth. And they have also something to offer, and we basically invited them as associate members to this consortium, and we are trying to, again, create these bridges between investigators, and they also contribute, but they have also some benefit of being a part of the consortium. There was one project that I didn't mention, that was Dr. Kwok Hong's project. He's now at NYU, New York University, and he's looking also, is a therapy project. He's looking at, he has definitely different animal models, and he's looking at the type of drugs that control the cell cycle. Again, quite a promising area in small cell lung cancer, and he's looking to basically to improve the outcomes with these, and to look at also the resistance that would develop to these drugs. Yes, so that's very important. I mean, all of us went through this stage where you seek advice from your peers. I think there are several types of advice that I can offer. They are almost common sense, but it's good to have this in mind. One issue that recently arose, especially in the United States, is the issue of reproducibility of data. It's very important in science, more than perhaps in other areas, that the data that one produces is reproducible and it's solid data. We know that science goes forward, and then there may be some discoveries in the future that may contribute or to expand the knowledge that we have. but those discoveries need to stand on a solid base. So my suggestion to young investigators would be to not to hesitate to go ahead and ask these tough questions, but once they have some data that they believe is strong data, I would suggest to them to verify it even more than they would like to think, and to look at it from different angles, so that once it's being published, once it's out there in the public domain, there is no doubt that the data is solid. Unfortunately, many times it happens that not by a mistake or neglect, but maybe not all the questions were asked, and then you end up with a situation that there isn't a published paper, but nobody can reproduce the data. So that doesn't look good. The other thing what I'd like to suggest, especially to PhD scientists who work in the lab, whatever your focus is, I would also try to make a link or try to get in touch with the clinicians who do have the clinical questions. I'm coming from a translational culture. That means that we sponsor translational research. And while basic research is very important, and there are some questions that are answered today, but their clinical utilization will be in the far future, maybe 10, 15, 20 years from now. It's always good to have an eye, what's the current clinical need, and try to modify it. Because sometimes, if you don't do that, you may happen, as some people did, that you work for 2 years on something that you feel is very important, and then you talk to the clinician and he or she would tell you, well, this is nice, but we have other means to handle this. So your work becomes to be redundant or not really important. So I would try to always check whether what you're working on is really relevant now or in the future to a clinical situation, because ultimately all of this is not only because we want to understand, we obviously want to understand what's going on in the biology of the disease, but ultimately I think it should be then used for the common good, and so that our patients have benefit, or even better, if we can prevent the disease, that we can do it in a meaningful way.